Okay, so I will introduce or talk a little bit about Medea for people that don't know Medea. We're a 501c3 nonprofit educational organization. Our mission is to support the region's technical, scientific, and industrial workforce by having programs like this on commercialization, development, business operations, and finance, and by having these meetings, bring people together and encourage collaboration and networking. We also, as Brenda pointed out recently, we got a grant to help with the BizStat Institute in trying to close the digital divide, bring broadband to this area of San Diego Valley. So Brenda's spearheading that. Our future meeting, our next meeting is November 10th. Still kind of TBD on the topic, but that's the next time we're going to meet, probably here. So look for that. If you have any feedback, or you can reach us at email@mediatech.org, so you can send us your comments or ideas for future programs. After we're done with this, we're going to go across the street to Cabrera's to have some refreshments. So if you're going to be here, and if you're still here and want to join us, that'd be great. That I will introduce Dr. Chris Busser. Chris grew up in the canton of Zurich, Switzerland. He studied biochemistry at ETH Zurich. He moved to the University of Ulm for a PhD in virology and electron microscopy. There he went to Berkeley to continue the same microscopy studies on different substances. I think it was yeast. Yeast. Yes. So he knows a lot about yeast. If you have any interest. And then he went to Cedars-Sinai to work on regenerative medicine of the spine. He also, along the way, founded an imaging services business called Dormant. And then he finally joined the faculty at Oak Crest, where he's been working on the imaging core and the startup incubator. He's founded, besides Dormant, he's also founded or co-founded BioCubic, which is another startup. And he's gotten several grants. Two of note is the U.S. Economic Development Administration's I-6 Challenge Grant, which is an incubator called, I guess, Most IP. He's also gotten a grant from the U.S. Small Business Administration Growth Accelerator Fund Competition, which is a grant for helping underserved entrepreneurs in the region. He speaks 3 different languages. I don't know if we'll share them all with us tonight. English, German, and French, and has 3 nationalities: U.S., Switzerland, and Belgium.
Yeah.
So
I'm collecting.
yes. So please welcome Dr. Busser.
Thank you, Bruce. So thanks everyone for coming. It's really, really nice to see that so many current and former Oak Crest students showed up. That's really, really great. Honestly, that always makes our day at Oak Crest when we see that what we're doing has an impact. So I'm gonna give you a really big picture overview. And I structured this really on the short side to leave time for conversation. So interrupt me at any time. Whenever you have a question, just make yourselves visible or ask. And the message really I want to transmit here is why Oak Crest is so different from other institutions in research and in education. And what it really is at the end of the day is that we essentially operate like a startup. See? There we go. So we are a 501c3 nonprofit, as well as Medea is. And we are really heavily focused on chemistry, biology, and biomedical research. But it's only part of what Oak Crest is. So while the research drives the nonprofit in general, we leverage everything we do. So typically, what we do first is we try to get an education piece into our research. So our research is NSF, NIH funded, so it's competitive federal research. And we use that to teach the hands-on piece to students, particularly in the lab. So we will always have questions that come up during our research projects that we can't easily answer, or we don't have the time for it, or it's a crazy idea that came up. And those are typically the questions that we then hand off to students. And really, it's a question that we don't know the answer to most of the time. Typically, we have a wild idea and a kind of a guess where it's going to go, but we really don't know. And that helps the research as much as it helps the education piece. And the third pillar of Oak Crest is really the innovation. So we develop a lot of technologies in the lab because of our research, because of our curiosity, and all the crazy experiments we do when we get the chance. And we've become better and better at translating those discoveries into products, into patents, and the whole infrastructure that we built around that, we're trying now more and more to make that available to outside, either companies, individual researchers, even pre-company formation researchers with a great idea who need a place to play around with their stuff. That's essentially where we want to be. And the easiest way to really describe Oak Crest is that we're a research university at the size of a 23-year-old startup. So we're not a research university. We don't grant degrees, but we do all the things that the university would do. And at the same time, we run the institute like it was a startup. And it's not a startup because it's already 23-year-old. It was founded in '98. But the structure of the institute is really very much like a startup. So it's a very dynamic, flat organization that can really pivot on very, very short notice. And this is who we are. So historically, we hover around 20 people. We have 5 faculty. Mark is our CEO and president. He's also an organic chemist. We have John, who is a chemist as well, specialized in spectroscopy, but he's also manufacturing, and he's a genius with equipment. So whenever a piece of equipment breaks, typically he's the one who gets called first. We have Paul. Paul is a microscopist by training, like I am. And Paul runs also the education arm of Oak Crest. I myself, I'm a microscopist, and I'm more on the business startup side of things. And we have Peter, who is an MD, so he's a gastroenterologist at UCLA, and he's our medical expert. So whenever we need help on the medical side and also on the regulatory side, he has an enormous amount of expertise.
What's Peter's last name?
Anton. And then we have Manji. Manji runs the microbio and molecular bio facility. And he's really, whenever it comes to growing bugs and doing biochemical assay, he's really fantastic. He knows an inordinate amount of stuff in that field. And then we have administration. We have Wendy, who does our books and make sure that the institute runs smoothly. Kirsten, who is our grants manager. And then we have the rest of the gang that basically takes care of all the day-to-day operations, and our research associates as well, who a big chunk of them actually are former students who come and join the team. And yeah, it's a very, very fun organization to be in. And as you can see, it's very flat. And while we are all, or most of us are scientists by training and do our own little piece of research, we also have management roles. So as I said, we run this institute. It is ours, right? It's the product of our work, and we're responsible for it, which really makes it, gives it that startup vibe. So it's never boring, and no 2 days are the same, but you're constantly running. It's a constant find grants, do the research, and find ways to really help the local ecosystem. And one important thing that I've realized over the years being at Oak Crest is that by doing this, by being this open-minded and interested, it's a magnet. Oak Crest, by its nature, attracts more of the same kind of people, which is really fantastic to see. So we don't have to go out there and look for people. As long as we communicate what we do, people understand what it is and join the team in one way or the other with the extended family. So as I said, we're driven by research. This is just a copy paste of, I think, some of the papers that we've published in the last 2 years. And when you look at it, there's a big chunk of it that is in HIV prevention. That is really our core expertise, or what we're most known for in science. Implants, essentially, to prevent the transmission of HIV, for HIV prophylaxis, right? So PrEP. And that really has grown. It's the underlying foundation of the Institute, and it has allowed the Institute to diversify into other areas. So there's another one, the COVID one. So we have, and I'll talk about that on the next slide. We have a COVID paper that finally got accepted. It took like a year or so to make it through the process, but it's out there. And we also collaborate quite a lot with outside investigators, particularly me on the imaging core. So the imaging core is essentially my first job, and the startup incubator is my second job chronologically. And a lot of the research that I do is collaborative research with either nonprofits, universities, or for-profits as well. And when you look at it from topics, so it's really chemistry and biology, and then everything in between. So historically, we've done a lot of air and water quality research, catalysis, and origin of life research on the chemistry side. The origin of life is largely in collaboration with JPL, kind of figuring out what chemistry you need to get to amino acids, which are the foundation for life on extrasolar planets or on moons within the solar system. So all the icy worlds research at JPL. And then on the biology side, it's microbiology, it's the environment, molecular and cell biology, things that we can access with biochemical techniques and microscopes. And those are basically the two ends of the spectrum. And then we have everything in between where those areas overlap, which is largely the medical device and infectious disease field, where you need the chemistry on the drug side and on the drug delivery side, and you need the biology to study what happens in that specific system. So we cover all of these areas, and we use that research we leverage that research for everything else we do. And that's the big question, right? Locally, what does that mean for our community? Because at the end of the day, these are your tax dollars and my tax dollars at work. So how do we justify all of that money that's being spent at Oak Crest? And it really comes down back to the three pillars of Oak Crest. One is the research. So the research funds an expansion of infrastructure and expertise. So we have now about 30,000 square feet of space with, I mean, endless, almost endless amount of equipment that keeps growing and keeps shifting depending on where the research pulls us. And this is now, that's basically what we're providing to the ecosystem, right? It is the extensive research infrastructure and our know-how that we then leverage to do education. As you've heard, we have we have two current and two former students here. It's really, this infrastructure gives us the ability to pull in students and really put them on high impact research projects before technically they would be ready, quote unquote, in a large university. They would essentially have to wait till late undergrad or grad school before they would touch any of these pieces of equipment that we have. And we have exactly the opposite drive. We put them onto these pieces of equipment essentially on the first day or in the first week. And that really gives them a huge leg up. They're at least a year ahead of their peers when they enter the undergraduate part of their career. And then the third part, again, is innovation. So essentially, we've figured all of this out for ourselves. So we had to put the infrastructure together. We had to figure out the regulatory environment, even, I mean, lab safety and all of that, biosafety issues, and then obviously IRBs, informed consent, all of that. And we can now provide all of that to basically the innovation space here in the region. So startups, large companies, small companies, individual researchers who think they have something and they want to test it. We're really trying to make all of that available to them. There we go. And what that means on a daily capabilities front essentially was really tested by COVID-19. And I have to say, this was just amazing to watch. So as you know, COVID-19 hit us at some point early 2020. We still don't know exactly when and how, but at some point it became a problem. And the way I remember it was really early March, when suddenly there was a spike in potential cases. We still didn't have testing capabilities. We didn't know what was happening, and everyone essentially reflexively just shut down everything. And we were extremely worried about that because, as again, a small institution looking at a shutdown for unknown period of time, 6 months, a year, whatnot, it would have been potentially existential trouble for Oak Crest to really close the doors permanently. So in typical startup manner, we put our heads together, and that's mainly Mark, Peter, and Manji who really spearheaded that. And I think within 3 weeks, we had an IRB approved testing study put together. IRB, so institutional review board. So essentially you need to have ethical approval to do research like that on people. And we got that together very, very quickly. Manji established the qPCR testing routine, and we started testing end of March. So really, when everyone was shutting down, we were cranking up and we started testing everyone at Oak Crest plus spot testing relatives 3 times a week. And we've done that since. We have not stopped since. So we've done now 1.5 years of testing in about, thinking about it, 1,000 tests is about 3 months. So you can do the math. We're in 5, 6, 7,000 tests performed at Oak Crest, again, with a staff of 20 people while still doing all of our other activities. So that was quite amazing to watch. And we published some of the results that I put there in the figure. So very early on, we recruited 1 participant into the study we knew was clinically positive, and that's the left panel there, A. And you can see these are qPCR results with dates. So it starts April 1st, and then it goes all the way to late May, early June. Blue means positive. So our test has 3 components. It measures 3 components of the virus, and a test will register as positive if all those 3 components show up positive in the qPCR test. So blue means this is definitely positive, right? Absolutely all 3 came back positive. Green are the inconclusives, means typically 2 out of the 3 turned positive, 1 was negative, and we call those inconclusives. We still think that there is something going on, but it's just at the limit of detection. And then the red dots are the negatives. And this was a realization very, very early on that now we have a much better grasp, but back then was like, oh, we have a problem, because if you look at A, that person was positive for more than 2 months, right? Almost 3 months. So how do you deal, and wildly positive, right? Those blue dots at the beginning are really, really heavily positive. So how do you deal with that, again, from a public health standpoint, right? We all assumed that 2 weeks, right? And then everything's fine, go back out there. But you have detectable amounts of virus for multiple months. So what do you do with that?
Chris, are most of the testing being done only on one determinant? If I went to USC before I had a procedure and they, swabbed me, and did they only test that against one COVID?
Typically it's 2 at least, again, to get confidence, but it varies. I forgot now what the standard clinical diagnostic test does. Honestly, I'd have to look it up. Okay. But you don't trust one single PCR result ever, right? You really shouldn't do that. And the diagnostic test, from what I remember, is also 3 components. They might be slightly different than ours. But we've essentially adopted what really was recommended early on by the authorities. So that was one, right? Highly positive for a long time. And then case B came a little bit later on in May. And that was what later was called asymptomatic. So positive, but when you look at it, the red line is essentially where you go from detectable to non-detectable, more or less. So just barely detectable, and would probably have registered negative with a clinical test. So our test, because we collect slightly differently, we can see 10 copies of viral genome, which is far more sensitive than the clinical test. So most likely, if this person went out and got the clinical test, would probably come back either weakly positive or negative.
What's the difference in the collection?
So if you go for the standard test, they have to preserve the sample in a large amount of buffer. So right, when you swab yourself, you stick the thing in the tube, you see that there's going to be like 1 milliliter of liquid in there. That's a preservative to keep the viral genome stable through transport till it reaches the lab where the assay gets run. So you have to stabilize the genome over that period of time. COVID is an RNA virus, and RNA is very unstable, right? It deteriorates fairly quickly if you don't preserve it well. And for us, that's not an issue because we collect and we run the test immediately. Within an hour, we're already isolating RNA. So we can save on the amount of collection fluid we put in there, which allows us to essentially at least a 10-fold improvement in the dilution factor. And that just pushes the detection limit down to the approximately 10 copies of RNA, which is quite amazing. I mean, when you think about it on a swab, you can detect 10 copies of virus. It's pretty amazing what technology can do these days.
So what would be going on a limb to say that because of the testing, we thought we were negative and we went out to the public, is that, would that not lead to more spread because you have a
Not necessarily. So how much virus do you need to get infected? We don't know.
No.
We still don't know.
We still don't.
We guess. We say, yeah, 1,000 is probably one, right? Yeah, maybe 10,000. Yes, you're probably going to spread it, but it's still a guessing game. We still don't know. Now everything has changed, of course, because this was the original strain. Now we're with Delta. Those numbers have changed. So all of this is such a fast-moving scientific problem that we're always lagging behind. Essentially, what we understand now doesn't necessarily apply to what Delta does. And really, it's experimentally, it's almost impossible to tell how many copies of virus you really need for a person to be infectious. There, again, you can kind of set boundaries in terms of probability, right? Technically, one copy, one virus is theoretically enough to infect you, but it's statistically improbable. And that was one of the major issues in all of this, in communicating all of this, is that at the end of the day, we're dealing with probabilities. And along the way, we make educated guesses that lead us to a point where we have reasonable confidence in saying that a person is contagious or not, but we still don't know for sure. Most likely, case B with that low viral load most likely was not contagious. It's unlikely that you would infect someone with just 10 copies of virus. Yes?
I think the other factor you have to consider is the person who's exposed, so B is low viral load, so less amount of virus, but then also whoever they're talking to or interacting with, this is why we're still wearing masks, right? Because you want to reduce even if the person has it. And also the third thing, their immune system, the strength of their immune system, so how quickly and how long. I mean, number 8 is amazing in the sense that how long it's, virus stay, in the system though. So I think that's the other thing that makes an impact is after the exposure, even if it's a low viral load, how strong is your immune system to be able to fight it back and how quickly?
How close are you? How well ventilated is the space, right? Are you on top of each other? Are you sharing food? All of that, and it just becomes an impossible task to estimate the probabilities at this point. It turns out that the 2 weeks quarantine probably worked, right? It seemed to get things kind of under control, so it's a good sign. But at the end, we don't know. What we definitely know is that masks work. And that is the message in all of this. We know that masks work. They're the easiest solution to this. And again, anecdotally, there is evidence that even if you're in very close contact to a contagious person, the mask protects you. There's all kinds of evidence for that. Yes.
Doesn't it have to be a special kind of mouse?
Yes, and now that's where we're getting into probabilities again, right? So the better your mask, the closer it seals, the thicker the cloth, that probability goes down and down and down and down. And it's N95, right? The 95 means 95% of particles in that micron range or submicron range make it through the mask. That is the standard. So you can say you've dramatically reduced the amount of virus that passes through a mask. It's never going to be 100%, right? But it's good enough, and it diverts the entire flow of the breath away from the other person's face. That helps as well. So, but it's always a probability, and that is the hard thing to explain to a non-scientist, is that there is no certainty, and people then take the absence of certainty as an indicator that it's just whatever it is. So that has been a real challenge. It's more in the communication end than on the science. The science has just been absolutely amazing. I mean, the worldwide output in COVID research was just mind-blowing. And that's why it took almost a year for our paper to get accepted, right? We published it on the preprint server, which is also a good change, actually, scientifically. The way research get disseminated like that has actually benefited from COVID.
So was it available before?
It was available before. It was used particularly in the physical sciences. They had the habit of already for many years to pre-publish on a preprint server. In the biological sciences, there was still the fear that it's basically you release the data before it's accepted by a journal. And technically in the early days, some journals actually said, if you pre-post that, we're not going to accept it because it's already been disseminated and it's not original research anymore. And that has completely changed. So now actually many journals, because of COVID, many journals now actually automatically, when you submit a paper, they will ask you, check a box to have it published on the preprint server.
Oh.
So this has really, it has, I mean, obviously the whole pandemic has been a disaster for humanity, but it has had a couple very, very strong benefits on the research side. Everything has become far more efficient.
What's the prognosis of coming up with something that will kill that virus?
Very difficult. There's obviously lots of attempts, right? We just have a pill now that's probably going to become available, but the message is it's always a lot easier to prevent the infection than to deal with the virus once it's in the body. A drug that kills the virus is much, much harder to develop. It takes a lot more safety research. It takes longer, it takes more money, so it's difficult from that standpoint. And biologically, it's far more complicated because you're dealing with the virus within the human body, with the individual cells, how they produce virus, the immune system on top of that. It's extremely complex. And each of us has a different immune system, a different immune response to it. So it's incredibly complicated. So killing the virus, if it happens, it's going to take some time. And of course, now when you look at it from a financial standpoint, as a drug manufacturer, by the time you've developed that drug, the pandemic might already be over. So it's a very tricky subject. Doesn't stop people from trying. There's a lot in the pipeline, but I'm not sure how helpful it's going to be. The vaccines are really what's going to make this pandemic end, not the treatment. Yes.
The answer might probably be no, but like, do you know if the COVID virus goes dormant in the body? Like,
Should not. Should not. And here's again the scientific uncertainty. Coronaviruses don't typically do that. It would be a huge surprise if it happened. At the same time, we've had other instances. Ebola was another one, right? Where when we had that Ebola outbreak in, what was that, 2015-ish? That large outbreak in Africa where it was, okay, it's Ebola, right? People die and then the virus just goes away and everything's fine. And then 2 years later, they found traces of virus in a patient in the eyeball. Because the eyeball is isolated from your immune system, similar to joints as well. So there's a chance that virus can hide in there for a long time, but we don't know. So again, this is uncertain. I would be surprised if it happened, but scientifically you can't exclude it.
But it can certainly live for a fairly long time, as that's evidence.
It replicates, yes.
So that long COVID is a real thing, and people do. Is long COVID virus?
We don't know. We don't know. We don't know. Long COVID might be a downstream side effect of the immune system, and it might be a very, very low level replication that is just impossible to find. And that is something the field of virology hasn't figured out yet. There are other viral diseases where there is a suspicion that there is continued replication at a very low level, but it's so low that you can't find it. Needle in a haystack, right? The human body being the haystack. So simply we just don't know. We really don't. And it's obviously it's a huge concern. One early study out of Chicago said that 30% of people admitted for COVID have long-term neurological symptoms. So it's, I hope it's not going to turn into a big issue, but with the cases, the case numbers, it's not looking good. Okay, so research, that was that part. Outreach. Interesting, somehow the formatting changed. There we go. So the other piece that we really do is outreach. So we work with the community and we start, as you can see, very early on. And I was tempted to put the text underneath there. And I just decided to put that picture in because that picture really doesn't need any text. That is what we do, right? It is showing a path to science, to STEM in general, very early on. And we know that the earlier we can get to kids with STEM concept, the higher the likelihood that they actually consider this a career choice and a field. And there circled are all of the programs that we've done historically. It's probably a little too small to read from there. But the Junior Researcher Academy was a big piece this year. We couldn't do it in person, but we did it virtually. And this was really, again, the best that we can deliver, because we as faculty, we were involved in this. We designed the whole thing, particularly Paul really worked day and night on this to put it together. But when the time came to actually teach, how many did we have? 20 kids over Zoom with science experiments that we had purchased thanks to a donation by Boston Scientific. So we bought kits. We gave the kids to the parents and the parents handed it to their kids. We then had the students, our students actually run the program, and Lisa and Elijah are here. They're 2 out of the 3 who ran the Junior Researcher Academy essentially independently. Well, really us as faculty, we were in the background to help if things somehow didn't work out. But it was fantastic because we could just lean back, and obviously they learned, our students learned to handle research, to plan the experiments, and then to communicate all of that, which is a huge challenge. It's hard for us. So it was fantastic to watch. And my daughter, Hannah, actually was in the program as well. So I was, while you were explaining things, I was sitting in the background watching you. It was amazing. So Hannah was, in the beginning, she was hesitant. She was like, oh yeah, not another science thing. And after the first JRA event, she was hooked, and she went all the way till the end, and she did every single kid. So really, congratulations to you guys. This was absolutely fantastic.
So a great way to learn is to teach.
Sorry?
I said a great way to learn is to teach.
Yeah, that's the best way, right?
And do it yourself, because I do think if he was just showing how to do it, it wouldn't have been as exciting as the kids you guys sent at home and they were doing it with you. Sometimes they struggle. The solar car did not work out very well. It didn't go to places like it's supposed to, but still, the failure of that car,
Yeah.
trying to put two solar, instead of one, which is what was instructed, doing all that herself, and then like you, I'm sitting, like she's on the kitchen table and I'm, doing my stuff and watching and doing. I'm a scientist at heart, so I can't help it. So that was really good. I'm very thankful. You guys did great.
Again, we've never done this before, and we were highly concerned because we had no experience in doing this over Zoom. So it was forced on us. But again, here as well, the pandemic really showed us that we can do a lot more by using digital media to really communicate science a lot better. So yeah, but it was amazing, lots of fun. We still hope that we'll get to the in-person event soon. And here's the part where really we get to the next level, which is teaching students. And again, this has been happening since '98. Really, Mark founded Oak Crest with this as the original idea: use science to teach and use the teaching to fuel the science. And you can look it up on our website. This is the alumni, our wall of fame, where all alumni who had a lab experience are listed, and it's a long, long wall, and it keeps growing. Pre-COVID, we were at the point where we had more than 100 students a year come through the institute and actually get the meaningful hands-on experience, which again was 20 people. You run 100 students through a summer program that lasts maybe 2.5, 3 months, while doing your research on the side. So you can imagine the madness. Essentially, every summer, Oak Crest would quadruple in size. And it was complete chaos, but somehow it always worked out. And that is really, for us as faculty, that is one of the most meaningful things that we can do. And it's really, I'm so happy that all of you showed up today, because we now hear, whenever we reach out, we hear from people who've been with us in 2008 and 2005, and they still remember and they still credit Oak Crest, the hands-on experience, as something that was really formative in establishing their science identity. And that really, this is a career that is accessible to everyone if you provide the right training and the introduction to it. It really makes an enormous difference. I mean, we have lots of students who afterwards tell us, and I never thought that science was actually a career that I could pursue. It's not something you normally think of, and it really, the hands-on piece, the moment you give the students the ability to actually do the research, it's typically the first month or so is complete shock. And I'm sure you can speak to that, right? You don't know where things are, you don't know what you're doing. You don't understand if you're in the right place, if your experiments are working, and it seems like nothing is working and you don't know where you're going. But eventually you learn that that's how research is, right? You never know what you're doing, and you do an experiment, you get data back, and you keep repeating that until you understand what's happening. So giving us as faculty the ability to teach that to the next generation is really, that's the best thing we do all day, really. It's just incredibly rewarding. Yeah, and here we are. Why does it matter, right? So you have school, and school is supposed to teach you all of that, and it doesn't, because school is still school. School, your work gets graded, and there is a right answer and the wrong answer and the grade at the end. And that's really not how science works. I'm sure you've learned that by now, right? You're used to having a right answer. You're somehow supposed to get to the right answer. And there just isn't. In science, there is no right answer. It is whatever comes up in your experiments until you refine and redo and repeat. And at some point, you start believing that the answer you keep getting over and over is probably correct. And really the point in this is it gives the students a way to establish their identity, right? It is really, I am a scientist. It's not I'm working with scientists, but I am a junior scientist myself. That is where the shift happens, and where suddenly there's a light bulb that goes off and, hey, I can actually do that. This is not just a theoretical lab course and something that maybe I could play around with as a hobby, but no, this is actually a career that is now accessible. And I really, really encourage you. I'm going to plug here at the bottom also our fundraiser. We're actually raising funds for our fellowship program right now. We have a promise for a matching contribution by a donor. And click on it. Even if you don't donate, just click on the videos and watch those videos. Yeah, it's amazing. It's unbelievable. And it's lots of fun.
What's the amount of money you're trying to raise?
Sorry?
What's the amount of money you're trying to raise?
As much as we can.
What's the matching though? Is there a limit on the matching?
There is a limit on the matching, but it's pretty high, so it should be fine. Again, you can imagine how much effort it takes. And typically, the problem is that we get research funds, but we can't use research funds to teach, right? Research funds are allocated as research funds. So we do this as much as we can on the side with all the funding we can scrape together. So really, money is a huge issue in the number of students we can put through these programs. And then the last bit, which is really where my heart is the innovation ecosystem. So anything that we've learned, we can make accessible to others. And that is not only the research itself and the research expertise, but how do you set up a lab? What are the safety issues that you're facing? Biosafety, chemical safety. Filippo and I have discussed that a lot in our incubator, right? Where does the power come through? How much power do you need? 220. How many lines do you need? Separate fuses? What kind of equipment do you have? So it gets very, very, very complicated very quickly. And typically, speaking of myself as an academic researcher, I never had to learn that, because in a large university, you have a maintenance team, they come in, they put it in, and it's there and it works, right? And then they walk away, and you never learn how to set all of this up. So it's a serious issue for academic scientists who have an idea and may want to try to develop the technology somewhere else, separate from their academic career.
So, quick question, you share the research or the findings of the research you have with your grants, when you're teaching? Can you share that?
Yes, we can. I mean, whatever we develop, we typically publish. So it's already out there. And certain things we do keep to ourselves if we have technology that we intend to develop and spin out. So like my spin out now, BioCubic, is stuff I stumbled upon doing Oak Crest research that I'm pulling out of Oak Crest and essentially developing within the startup. So sometimes we do keep things to ourselves, but our general research, whatever is NIH funded, we're required anyway to publish. So that's out there. So the side projects are typically what we develop internally. But again, this is something that we're just exploring now. We are essentially sitting on a gold mine, right? Oak Crest doesn't have a tech transfer office like the large universities. So we have
somebody with expertise right here.
There it is, right? We're not used to thinking that way. We are used to working in our grants, do our education, publish great science. And then sure, some of the things we try to develop, but it hasn't been a focus so far because we lacked the infrastructure and we lacked also the legal aspect of it. It was, right, how do you, as a non-profit, how do you spin out a for-profit and make sure that the non-profit doesn't get in trouble? So you have to make sure that everything is done right, is done properly. So that took a long time and a lot of effort to develop, but now we have it. And we're actually capable to really take technology all the way till the end to clinical trials. And we have ourselves on the HIV prevention front. We've just completed the phase 1 clinical trial that was the vaginal ring for HIV prevention. So that is an Oak Crest technology that was developed in-house and brought all the way to phase 1 with NIH funding and of course with a large group of collaborators that contributed to the whole project. So it's always, right, with 20 people, it's always a collaboration with others. But we can do it, we figured it out, we figured out the manufacturing piece as well. We know our way around IRBs, again, ethical approval. We can also work with the FDA. So we have FDA submissions. So we know what is required to do that. And we can offer all of that to a startup. We have to figure out how at the end Oak Crest gets compensated for all of that hard work, but it's possible. And that really allows us to work with any startup pre-formation, so even just the concept, the idea, all the way to a late stage development of devices, drugs, testing of drugs, and so on. And we've done that now, even through the pandemic. We have, that's the part down here. So since 2019, when we got that EDA grant to build out the incubator has supported 13 startups, has been on 15 SBIR submissions, and the startups raised more than $6.5 million in combined funding. And that is basically while we were all shut down and waiting to see what would happen. So we have companies that are completely virtual. They use Oak Crest essentially as a CRO. So they send us stuff, we do the research and we send data back, and they use that then to apply for SBIR funding. We're also helping on clinical trials to do analysis and so on. And then we have the very early stage startups that just need a lab space on short notice with a short runway to just see if the concept has legs or not. And if it doesn't, they have to be able to fold within 6 months. So we have the whole spectrum, and we're just getting started. So these numbers are gonna explode fairly soon. I mean, just this past SBIR cycle, we have submitted 4 applications. And again, really, it's just the beginning.
And how many was granted or are you waiting?
Waiting. The scientific review is happening October, November. And then funds, then there's final committee is, I think, January, February, and then funds start in March. So we'll know fairly soon, but it's looking good. And we're already, again, with SBIR, you can't wait for the cycle to complete. You already have to start with the next technology for the next cycle, and you just keep going. The difficulty is that it takes so long for funds to arrive, right? You're essentially waiting from a submission window in September till in March, the money arrives, if you get it, and your success rate somewhere around 25%, which is pretty good, but it's still more likely that you're not going to get money. So it's hard for startups to plan that way. And so our offer to essentially start establishing things virtually is very, very meaningful for those startups, because they don't need any infrastructure. They just essentially they compensate us for reagents and labor, labor being the biggest factor in those projects, but it's a lot cheaper than getting lab space. And here's just a collection of all the collaborators, partners, funding agencies. The city of Monrovia as well has been instrumental in all of this. They've been incredibly supportive from the moment Oak Crest moved in 2016 to Monrovia. So Monrovia has been fantastic for us. It has allowed us to do things that we were not able to do before. And we've really developed a very, very broad network now, locally and nationally with our scientific collaborations. And now the awareness that the EDA and SBA funding has given us really put us on the map nationally. And we have now a pretty good network as well on the commercialization side. It's growing, but it's looking great. The future is really looking very, very bright. So spread the word, let other people know that we're here, and help us to basically become more seen and more known locally, regionally, and nationally. And that is really, these days, it's all social media, YouTube. It's really, it makes an enormous difference for us if at this early stage, people spread the name Oak Crest wide and far, because it brings unusual collaborations, to say the least, and it brings very, very interesting projects to Oak Crest from people we didn't know and projects we didn't know existed. So it's, again, very important, but it's also very fun. And with that, I'm gonna finish, and thank you all for being here. And let me know if you have any more questions.
Dr. Buser?
Yes.
Hi, Agabrayo again. Sorry.
Hi.
Hi, I actually wanted to ask a question.
Sure.
Okay, so I did an internship with you guys, maybe, I think it was like 11 months actually. I worked with you for a little bit, with Dr. Paul Sr.
Right.
and Manji
Yes,
for a while.
I do remember.
And yeah, thank you. I think I did sectioning for you, which I'm really bad at. So I actually transferred to ASU, but when I went to ASU, they basically send us all back, right?
Yeah.
So we had to go full online format, and actually ASU has a new online biochemistry format that I'm actually currently in. So I chose that to, continue my education. But in the sense that pandemic happened, this is all new for us, going fully virtual. Good thing, I'm grateful for Oak Crest, because I was able to get sufficient lab experience to be able to be competitive. But in the essence of education, do you think that Oak Crest can move into a partial internship online? In the
We
sense
are
like you get,
working on the, yes, we are working on the online portion. The problem is, how do you teach that, right? At the end of the day, what we're really selling is the hands-on piece. That is what we're good at. So transferring those skills online is very hard. Plus, honestly, the online piece is not our area of expertise. So what we're trying right now is to work with partner organizations to help us bring that online. What we are hoping to do is hopefully eventually to come out of this whole COVID mess, and then we can start again. The limitation that we currently have is that we're still testing, right? Oak Crest still operates under COVID lockdown. We do not allow outsiders who are not on our testing schedule into our main facility. That excludes the incubator. The incubator is up and running. But our main facility, we're still hesitant with Delta. It's still too dangerous for us to do that. And we can do that with our current fellows because we have enough funding to put them on our testing schedule, right? When you think about it, each test, if you went out there and got the test, it's somewhere around $200 plus minus, which includes the reagents and the labor. Just the reagents per test are somewhere around $40. So you can imagine what it would cost us to bring 10 students in just for 2 weeks, where we couldn't do much. That's currently what's limiting us. But the online piece is definitely something we're trying hard to develop. And that's also the activity. If you click on our YouTube channel, that's the motivation behind that, right? Transfer as much as possible online. But if you are interested in helping us with that, anytime, right? It's collaborations, right? Certain things we just cannot do, we're not good at, and we lack the skills. So if we can collaborate, absolutely, happily.
yeah, well, being online based, you learn a lot of data analysis and a lot of art. So that's always a different aspect of viewing. Yeah, because that's actually one of the projects we had to do is analyze, well, it wasn't COVID case, but the projection was crime rates in the United States, right? So one of our classes was based off analyzing homicides in the United States using R. And you're basically able to work in a remote area, but you're still analyzing data.
You want to run a course through Oak Crest?
Oh, let's see what we can happen. I mean, I wouldn't mind.
Perfect. Let's do it. So you see, those are really things we're absolutely open to. We'll help with all the infrastructure, creative ideas. Really, Oak Crest is a place where the crazier the idea, the more interesting it typically is, because it's a paradigm shift most of the time. So if you can come up with something, we'll definitely support you as much as we can. So collaborations, any day, anytime, whatever you need.
Thank you very much.
So if everybody was vaccinated, could they go in? And did you resume if people showed that they were vaccinated into your office?
We're just hesitant with the breakthrough cases, and we have employees with really high at risk family members at home, and it's too dangerous still right now. It's just too close. I honestly, it's a big issue for us as well. Really, this slows us down so much, but it's an impossible decision, right, to make.
It makes me worried. Like, you people are informed, and you're not doing this. I work somewhere where it's like, as long as you're vaccinated, you can go in. And I'm like, I mean, they would still have masks, but.
Yeah. I can't give you a good answer to that one. again, we have those discussions every week, essentially.
Yeah.
What is the risk profile? When do we stop? At some point we have to stop, but what then, right? What if we do get a case? Do we then shut down everything again?
So you're still doing all the testing, even though, I mean, I'm guessing.
We still continued because we were worried about the breakthrough cases, and we wanted to see, honestly, this is for us scientifically also very interesting, right? What if we get the breakthrough case? How does that look like? Where do they score on the viral titer, right, on the graph, on the qPCR graph? And what I didn't show is that we also have antibody data. So we all volunteered to give blood. I gave blood as well. And it was very interesting. Even early on, it looked like a significant number of Oak Crest employees had antibodies, even though they tested negative. So there's a lot of science that goes on that we are really, really interested in. Sorry?
That was pre-vaccine.
That was March 2020. So before it really became an issue. The virus was spread, obviously the virus was spreading for many months before that, but we couldn't test, there weren't enough tests, so who knows, right? But yes, I think about that on an almost daily basis. It really, particularly on the education front, it's a huge issue. Because we would love to have, and we get requests for 10, 20, 30 students, 5-year-olds, community college students, the whole range, and we can't do anything right now. It's incredibly frustrating.
Okay, well, we'll let you take a break. It's kind of more than an hour.
Yeah, sorry, way over.
But I think it's a lot of very, very fascinating, tremendous progress obviously being made, so.
Thank you.